Welcome to Telling the Tooth, the official mental dental podcast. I'm your host, Dr. Ryan Gross, and I'm joined today by two very special guests, Dr. Laura Weyrich and Kevin Ketagoda. So, for a brief introduction, Laura is originally from South Dakota and received her undergraduate degree from South Dakota State University. She then received a PhD in microbiology from Penn State. She then moved to the University of Adelaide, Australia, to study ancient bacteria that live in the mouths of our ancestors. She started her own research team there and led the first team to reconstruct microorganisms that lived in an extinct species, Neanderthals. In 2019, she moved back to Penn State as an associate professor in anthropology, bioethics, and the Huck Institutes of the Life Sciences. At Penn State, she leads research constructing microbial communities, or microbiomes, in people of the past and the present all around the world. And she's working with a team at the University of Adelaide to develop a new exciting technology that may improve oral health one day, oral microbiome transplants, which will be the topic of discussion in this podcast. Super exciting. And Kevin graduated with a Bachelor of Science with honors, majoring in microbiology, and currently in the last year of his PhD training, also at the University of Adelaide in Australia. He has a background in microbiology, biochemistry, and immunology, specializing in using in vivo animal models to study bacterial infections. At the present, he is the technical lead of the Oral Microbiome Transplant Project, which is his PhD thesis. In this project, he's developed and validated a 3D printed in vitro or laboratory system and grown dental plaque from healthy humans using these systems. Additionally, he utilizes a wide range of bioinformatic techniques to investigate the microbial communities grown via the 3D printed laboratory systems. Laura, Kevin, thank you so much for being here today.
Thanks so much for having us, Ryan. We really appreciate it.
Yeah, of course. Well, I guess we could start with the kind of background of this project. I know it was briefly mentioned a little bit in the introduction, but I'd love to hear directly from both of you, kind of how did this project start and what's the story behind it?
Yeah, I'll tell you a little bit about how we started. So during my postdoc, I started a big project to reconstruct the types of microorganisms that lived on the teeth of our ancestors. So we essentially used a time machine by using ancient DNA to go back in the past and reconstruct the types of microorganisms that people had, and even other species or other hominids had. So we've looked at things from Neanderthals, from chimpanzees, from bonobos, and all sorts of things. But one of the clear things that stuck out was that many of our ancient ancestors had much better oral health than we did. And in fact, they had completely different types of microbes that lived in their mouths. And that a lot of the, dental decay, the cavities, the caries, and the periodontal disease that we experience today in modern societies was actually something that came about only when we started practicing agriculture, which in Europe was about 1,700 years ago, but that differs all over the globe. And really, in studying that information, I was really frustrated that we couldn't do something about that in living communities today. How do we take that information and translate it into actual health benefits? Really, my husband was asking me a question one night, like, it's really great that you're understanding how we got into the current disease crisis we have, but when are you going to do something about fixing it? And so I wrote a grant application that was funded by the National Medical and Health Research Council in Australia, which is kind of similar to the NIH or the National Institutes of Health here in the United States. And they funded our crazy idea to essentially take donor dental plaque from ultra-healthy people, grow it in the lab, and then use it to transplant, hopefully one day, into recipients who might need it. These recipients might be people who suffer from really severe dental decay or really severe periodontal disease. And we hope that by transplanting healthy microbes, that we might be able to improve health. And so we've been working on this project for a couple years, and Kevin is one of the amazing graduate students that's been helping on the project and really running everything in the lab. Kevin, do you want to tell him a little bit about how you got involved in the project?
Yeah, so I'm the technical lead on the OMT project. So basically, my entire PhD thesis is on the OMT project itself. So like I said, you've mentioned my background as well. So my liking to this project came from So I've studied single infection models, which means one species caused a certain type of infection, but clearly that is not the case in reality. Always there's multiple things happening in the background to cause an infection. So I saw this ad, which basically was looking for a PhD student at University of Adelaide. And then I put my hand up, applied, got in touch with Laura, and moved interstate essentially to start my PhD in this project. And yeah, that's pretty much what my PhD's been on.
Fantastic. It sounds super interesting. I can't wait to dive more into the details here and what you've discovered up to this point. Just out of curiosity, Laura and Kevin, how are you able to find those ultra healthy patients used for this project?
Yeah, that's a great question. I mean, I really do hope someday we're sort of reconstituting Neanderthal oral microbiota and putting them in people because Neanderthals had way better health than we do. But in the meantime, because the bacteria at Neanderthals are all dead, we have to rely on what we have in living people today. And so what we did was put out a call in Adelaide for anyone who, for example, has never had a cavity, who doesn't have periodontal disease, and otherwise doesn't have any other systemic diseases that they have to take medication for on a daily basis. We brought them in to the lab. It's a dental research lab, so you can imagine a bunch of dental chairs all lined up, ready to go. And we did survey them out to make sure that was indeed true. And then we collected a bunch of information about their diet, their hygiene habits, their lifestyles, where they're from, and really used that information to then describe what drives healthy microorganisms in the mouth of people who are, these amazingly orally healthy people. I'm not one of those folks, I wish I was, because in fact, many of the people that we recruited are people that have never had a cavity, never had periodontal disease, never gone to the dentist, and never brushed their teeth. These are kind of, these unicorns of people that are out there. We also had a subset of other folks that, they had had a couple cavities when they were a kid, but otherwise hadn't had cavities in many years, and they don't have periodontal disease and had really great oral health. We also enrolled them into the study. But we found that, usually those folks were people who had crazy good oral hygiene. These are the folks that brush their teeth 4 times a day and do lots of flossing. And, we may have been recruiting next to a dental school, so I think we may have picked up quite a few dental students, which may explain that. But it is really great to know that there's different ways that people can be orally healthy. And in fact, a lot of our research of these ultra healthy, orally healthy folks really showed that there's not one single way to have healthy organisms in your mouth. Maybe you have really great hygiene and that contributes to it, but maybe you just have naturally really great microbes and you don't need to brush and you don't need to go to the dentist because your microbes have never caused you any problems. And those are really the people that we're after. I've also had people say, well, these must be the folks that are, vegan or they eat a really healthy diet. And that's not true either. We found quite a few folks that, again, never had a cavity, never been to the dentist, never brush, and they also eat tons of sugar. I mean, we had some people eating over 300 milligrams of sugar a day and still are perfectly orally healthy. So we really kind of bucked much of the current wisdom, I would say, when we were recruiting these individuals. But we're really looking for these unicorn type people, that don't have to go to the dentist, don't have to brush, yet have great oral health.
Yeah, that's really interesting. Kevin brought up this idea of the multifactorial nature to a lot of diseases, and caries, as you're talking about, is certainly one of those multifactorial diseases where you have the agent, the bacteria, you have the fuel source, which is traditionally the simple carbohydrate, and then you have the host, which is the two structure, which can be cleaned or not cleaned, and then some combination of those things contributes to the incidence of caries, and you're really targeting that agent part, the microbe part, which is, I think, a bit untapped. And so that's why this is so fascinating and so exciting to talk about. So in terms of this project, what are some of the preliminary things that you've started to see? Or maybe we could even start with, what's the experimental setup look like for this project?
Yeah. So once we recruited these 100 ultra healthy folks, we really wanted to then start growing their microorganisms in the lab. And Kevin is the guru when it comes to growing microbes in the lab using these in vitro systems. Kevin, do you want to explain what those in vitro flow cells look like and how we create them?
Yeah, so this is a 3D printed lab system. So first part of my PhD was trying to develop this system which is biocompatible. So we needed to make sure that we found a material that is biocompatible and it could be sterilized using autoclaving. But also it was resistant to chemical sterilization as well. So we have a name for this. So this is the flow cell, which is on this black sort of surface, which is a heat bed, so we can regulate the temperature. We usually set the temperature to around 37 degrees. And this bottle over on your left side is a media bottle. So we feed in media into the actual flow cell through these tubes. And the media is what we call artificial salivary media. And it's got a bunch of nutrients, and I will list them in the podcast so you all can go through it. And what you see in the background is a pump. So that helps pump the media through the tubes and into the flow cell. And the syringe is what lets us actually inject the plaque that we collect from people. The white little dots that you see in the middle of the system, they are hydroxyapatite discs. And as you all know, hydroxyapatite forms human enamel. So this is essentially, in a way, an artificial mouth model. And the bottle on your right-hand side is the waste bottle. So whatever waste that is collected in the flow cell goes through the last bit of tubing and gets collected into this waste bottle. Yeah, and this is essentially our system. And one of the reasons why we developed this system is to mass produce the dental plaque that we get from these healthy donors, because what we can actually collect from a donor is very low in biomass. The other thing that this system does is when we grow human dental plaque in a lab system, it eliminates viruses. So when in the downstream, when we're going to be doing an oral microbiome transplant, you want those harmful viruses not to be present. And by mass producing this plaque as well, we are able to form a biobank, which we have already done as well. So there's a few advantages to using this model that we've developed. So yes, we'll put up a picture of this in the podcast so people can go through and ask questions if they need to.
Yeah, that's great. Thanks for showing that, Kevin. I'm going to activate my inner tooth nerd for a second here because you mentioned hydroxyapatite, which a lot of the people who watch my videos do in fact know about. You're correct. Now, I'm curious because I didn't mention when we were talking about the multifactorial nature of caries, I didn't mention fluoride, which is one of the biggest defenses that we can give our teeth to stave off caries. Is the hydroxyapatite you're using kind of plain carbonate substituted hydroxyapatite, or have you considered using like a fluoride substituted hydroxyapatite or fluorapatite model?
So the one that we use is the plain carbonated hydroxyapatite. One of the main reasons for that is if you use the fluoride ones, it will potentially have an antimicrobial effect on the plaque that we're actually growing. So the plaque that we collect from people, you want to be mass producing it, which matches what you actually take from the actual person. So when you mass produce it, and if you have fluoride in there, it will potentially kill off key species in that oral microbiome community, and that would change the transplant. So we don't know what kind of effect it has. So if you take 100 species from somebody in their dental plaque, let's just say that, and grow it in the actual in vitro system, having fluoride in there might reduce that down to maybe, let's just say 10. So, which is what you don't want. So that's why we've used the plain carbonated hydroxyapatite discs.
I should mention that this 3D printed flow cell system was really the brainchild of Dr. Peter Zilm, who's at the University of Adelaide, and he's just done a tremendous job. Also, he's another co-PI on this grant, as well as Lisa Jameson, who's also at the University of Adelaide. And we have another PhD student, Sonia Nath, who did her doctoral studies on this. So, without their support and without their help, we wouldn't obviously be be testing any of this stuff. But this flow cell really makes it easy to take that donor plaque and mass produce it, but really be able to interrogate it and test it and push it and clean it in all sorts of different ways that we might be curious about. And so it creates a plaque that might be safer to transplant into people, and also creates one that we can sort of specialize in the future potentially to really make sure we're matching donor to recipient. So after we get done growing this plaque in vitro, then what we want to do is be able to put it into a recipient. And there's quite a few different ways we've discussed about doing this, but I think the best idea is to actually mix these microorganisms or microbial communities, or the microbiome, whatever you want to call them, into a hydrogel. And that hydrogel can then be painted on the teeth. It can also be put into, dental trays and sort of sat on the teeth for an extended period of time. And then we can sort of see how well those microbes then adhere to the tooth surface and sort of get moving and form that biofilm that we're really after. So there's lots of different processes that we can look at from there. And so far, we've just been testing it in rats.
Great, great. Let's maybe talk about some of the preliminary findings that you've had. What have you found so far in the rat model that you've been working with?
Yeah, sounds great. So we're really excited about some very preliminary evidence we have in rats showing that OMTs can be effective against the prevention or can be effective in preventing caries. So what we've done in this model is essentially take Sprague Jolly rats and feed them a highly carcinogenic diet, which Kevin can explain to you in a second. And some of those rats, too, we also infected them with Streptococcus mutans from humans as a way to ensure that those rats would develop caries. We then, in half of those rats, gave them an oral microbiome transplant. And so we were able to sort of give it to them on 3 consecutive days. And we were looking to see whether or not the OMT could help prevent caries development in these rats. And indeed, that's what we saw, both in groups that just had the carcinogenic diet or in groups that had the carcinogenic diet plus strep mutants. We were able to see a significant reduction in the amount of caries 5 weeks later. And that for us is just incredibly exciting. We are over the moon. We really didn't expect that. We also tested 2 different donors. And we developed a donor selection tool. So we were really after, donors that we thought had phenotypes and had, the types of characteristics that we were looking for that might prevent caries. So for example, the ability to buffer acid in the mouth. And so those were sort of the donors we selected. And with both donors, we were able to sort of see that significant reduction in caries. And, we're over the moon excited about it, but it's obviously very preliminary. And it honestly sometimes raises more questions than, provides answers. But Kevin, you should tell them a little bit about just the rat model and how absolutely, painstaking it was to do that work.
Yeah, so I'll just start with the diet essentially. So we fed them what we called a NIH Diet 2000. And this diet, the main ingredient of that is 50% caster sugar. And once we made that diet into powdered form, so how we did that is we sifted it through a specific sieve and then got it into this really fine powder. And we fed these rats essentially 5 times a day, every day for 12 days straight. And that's to induce caries. And the other thing that I think Laura mentioned was we gave one group S mutans, and obviously everybody on here should know that S mutans is one of the leading causes of dental caries. We grew that in the lab and we injected that. We brushed that onto the teeth of the actual rats themselves. So when the OMT was done, the hydrogel was again transplanted using an actual pixter brush. So we were there, we had the rat mouths open, and we just brushed the OMT onto the rat mouths. And we did that for 3 days straight. And this was to ensure the transplant was taken up by the rats themselves. And just before the transplant as well, I need to mention that we did do a chlorhexidine clean, so to mimic what is done clinically in order to sort of clean the oral cavity. So again, just literally there with a syringe and squirting it on and then just brushing it and just making sure we rinsed it. And there was no guidebook as per se for this. So it was all sort of done, sat down and talked about and done sort of without any previous knowledge. Yeah, I think it ran for 12 days straight, and throughout the entire experiment, we took fecal samples, we took blood samples, and at the end of the experiment, we took the rat skulls and we did micro CT scanning. And that's where the caries scores came about. So we had an actual dental practitioner, so Sonia Nath, who's also on the project, doing the public health analysis. She analyzed the micro CT images to score the actual jaws for caries. So that's where we found there was significant difference between the OMT transplanted group versus the non-transplanted groups. We also took things like serum, like I mentioned, and that's to measure for inflammation markers, to see if the OMT itself caused inflammation. We are still waiting on the data for it, so hopefully when we do the Q&A, we will have the data for that. We did take DNA samples as well, so we've done sequencing for that. We're still awaiting on the data, and we are really excited to see how the microbiome changes after the transplant as opposed to before the transplant in the OMT groups and the non-OMT groups. We also took histology samples as well. So we looked at things like gut tissue to see if the OMT transplant actually had a downstream effect on the gut. So we took quite a big number of samples to make sure that this transplant had ticked all the safety boxes as well. And it looked like it did. So yeah, so that is very exciting data.
Mm-hmm. Great. That's certainly one of the trade-offs to state-of-the-art research, right? You have this new revolutionary research, which is super exciting, but to your point, you don't have this model to follow. So you have to come up with the materials and methods sections not written for you. So you have to really be creative and thoughtful with that procedure. And it sounds like you spent a lot of time with those rats. That's for sure.
Yeah.
Well, at
It
least they
was
must.
literally 5 a.m. till 11 p.m. in the night
Oh my
every day.
goodness. Well, at least they must be a little bit happy to see you because you're feeding them 5 times a day, this sugar heavy diet.
Yep.
So maybe that plays in your benefit a little bit, I guess. Have you found with those preliminary findings, have you found, or I should say, with those healthy models that you've used for the transplants, have you found any specific bacterial species that seem to be playing a role in that?
Yeah, that's a great question. We are really just starting that process to start to look at individual strain and species variation at a genomic level. So far we've done what's called 16S ribosomal RNA sequencing or amplicon sequencing, and that allows us to just detect who's there. It's not really sort of what they're doing yet. We do on the sequencing machine right now, though, have some of the genomes for some of the Streptococcus species that have been isolated out of the mouths of these individuals. I have a hunch that maybe some of these Streptococcus species can't produce as much acid as we maybe think they might be able to in other people, for example, who study or who suffer from severe caries. So we're really interested in studying the differences at the functional level and at the genomic level across these species in both ultra healthy donors versus people, for example, who suffer from very severe caries. So, I reckon that there are specific taxa that will be really involved in this, but I do want to reiterate the fact, though, that we really do have to move away from thinking about one pathogen or one, microbe of interest and that causing disease, though. Really a lot of the microbiome research over the past couple of years has really shown that these common oral diseases like caries and periodontal disease, they're polymicrobial diseases, right? They're going to be caused by several different species working collectively and working together to not only, cause a disease, but create an environment that supports those pathogens to do their sort of job. So if we, I think, are going to use, this concept, we really have to be thinking about entire teams of microorganisms causing disease, rather than just sort of, one microbe, one pathogen, one disease. Strep mutans produces a lot of acid. That's great. But it has to be supported by species like lactobacillus and Veillonella to, in order to do that. And so we really need to be thinking about transplanting whole groups and teams of microbes, to go to war and to provide us healthy mouths, rather than sort of just one taxa. But, we're still really interested in what those individual species or taxa are doing. And we're excited to look at the genomics and functional information to figure that out.
So we've talked a lot to this point about the incidence of caries as the disease of interest. Have these preliminary findings shown anything with regards to, say, periodontal disease or any other oral or systemic diseases?
Yeah, that's what's up next, really. So we've started this with caries, because we think that there's some really clear pathways for preventing caries in the mouth using OMT technology or oral microbiome transplant technology. We like to shorten it to OMT because it's quick and fast, less of a mouthful to say. So actually one of the next experiments we're going to do is to try this in a periodontal disease model in mice. So we'll move from rats to mice and look at a different oral disease. But I'm also really excited about the potential OMT may have for systemic diseases. We know oral microbes, if they get elsewhere in the body, can do really nasty things. I think the best example of that is, colon cancer. With Fusobacterium nucleatum is in the gut doing horrible things, right? You can end up with colon cancer. So I'm really curious to see what that looks like. Obviously, that's like a, lifelong goal to figure all of that out. But I think if we don't start somewhere, then we won't ever know.
Yeah, absolutely. Well, maybe we could shift a little bit to the practical applications of this research and this technology in dental practice. And one of the things that concerns me as an orthodontist is the do-it-yourself dentistry model. There's a lot of teledentistry companies out there, for example, who will send patients impression materials via the mail. Patient will do their best to get an accurate impression, send it back. Then the company sends them clear aligners to straighten their teeth. This is without any radiographs, without any clinical exams, without any face-to-face doctor time. And bad things can happen without doctor supervision. I've seen it firsthand in the orthodontic model. So how involved do you anticipate a dentist would be with this technology with a patient?
Yeah, that's a really great question, and we've thought very long and hard about it. I think this technology can't be successful without the support of dentists and without their integration into the management and assessment of this technology. In fact, one of the first ways we'd like to develop this technology is by providing an option for dentists to apply in their clinic. So we envision this kind of looking like an additional therapy that dentists could offer maybe after a cleaning, an annual cleaning or a biannual cleaning. You might finish your cleaning and your polishing and really then be asked, do you want a fluoride treatment or do you want an OMT treatment? And depending on, what you're susceptible to, they might be able to select a particular donor out of the freezer and away you go, right? Mix it with some hydrogel, put it on some trays, you sit there for 15 minutes and you go home. That's really what we would, hope as sort of the awesome, potential goal that we might be striving for. I do think it's important, though, to think about other ways of getting this technology out to folks. We know there's a lot of folks, for example, that don't access dentists, even when they need to, either because they don't have financial support to do that, they don't have, access because they're in rural communities, or they're terrified, or whatever that looks like, right? It doesn't matter. And so there may be some way of providing OMTs, in ways that we might have to use sort of, teledentistry or something else to make sure that this can be equitable. Something I care about very passionately is making sure that we have equitable access to this technology, and that we can use it in ways that, really support communities, especially that are struggling to get dental care that they might need. It could be possible that OMTs provide a long-term corrective sort of strategy for preventing caries. And so, if there are populations that can't get to the dentist, this might be a really awesome, sort of tool to help, eliminate sort of downstream health issues that might come about because they're not accessing the dentist and they don't have access to that. So, I work a lot with Indigenous communities, both with Aboriginal Australian communities and folks elsewhere. And I think a lot about this sort of model in that context. We've thought about it both in the way that, maybe you would go into a pharmacy and buy a kit, and then you might sign up online and that would connect you with a dentist or connect you with somebody, or maybe you can use your phone doing that, right? And then we could ship you the microbes that you might need, that would be the least risk possible. Another model we've thought about, and we've been writing about this and we're ready to publish it, is maybe even just doing this in mobile dental health clinics, right? It might be that, hey, on Tuesday night, anybody can come to the town hall and get an OMT transplant of microbes that's, for your local community. And then we'll go on to the next town the next day. Obviously, there's, these are kind of pipe dreams, but they cause us to really think differently about how we're developing the technology. And I think that they really put us on a course to think about, different paths for commercialization, as well as different business ethics when we're developing companies to help promote the commercialization of this technology moving forward.
That's fantastic. I think I wish every teledentistry company had that approach, that it wasn't primarily for profit, but it was primarily for access to care. And so you've obviously put in that thoughtful consideration of that, and I think that's fantastic. As far as we talked about this before with the fluoride, and you mentioned that, a patient could get the OMT or the fluoride treatment. And I suppose having the fluoride treatment would potentially negate the effects of the OMT if it has that antimicrobial property, right?
Yeah, it's a great question. And honestly, it's going to require some research to figure that out. I think it's also interesting though that most folks living in industrialized countries, like the US or in Europe, a lot of us have, but not everybody, but a lot of us have exposure to, fluoride in our water or in our toothpaste. And so it's not that it's completely it won't be a completely new thing that these microbes are exposed to, right? Now,
Yeah.
when working with indigenous rural communities, it might be something very new, right? That their microbes haven't yet seen. So I think we have to take it probably on a population basis, but it certainly is going to require experimentation and for us to sort of test that out. It might be that we actually use fluoride as part of a plan to, kind of eliminate or reduce the recipient's microbiome before they would receive an OMT. Kevin talked about using chlorhexidine in the rats, to kind of limit their current oral microbiome before we did transplantation, did an OMT. And I think we need to think about that as well in humans, and we'll be testing that as we move forward, right? It could be that, it's a full dental cleaning, and then it's, a chlorhexidine mouthwash, and then it's a chlorhexidine sort of treatment directly on the teeth. And, maybe we think about some longer scale treatments or something that happens on a per weekly basis for a month or something, right? We don't really know what that looks like to then ensure that an OMT can be as effective as possible in that particular individual. So I think fluoride's part of that conversation that we need to think about and really be careful too, that once we transplant OMTs into folks, that we aren't just turning around and obliterating them with fluoride
Right.
is also another really active area of work for us. And so, it might be that you don't brush your teeth for a week or something. I don't know, right? Kind of sounds crazy, but those are the sort of things that we're going to have to look into to make sure we're supporting these healthy microbes and not accidentally turning around and killing them.
Right. Right. Now, one thing we didn't talk about so far is the safety of the OMT. Is it safe to use?
Yeah, safety is certainly a key concern of ours and something we've been looking at a lot. So in this rat caries model, Kevin mentioned that we're taking samples like things from like samples like blood or tissue samples, soft tissue samples, as well as gut microbiome samples. And we're doing that to make sure that we're not altering, sort of the healthy status of the animal and seeing what that might look like in humans. So we've been looking, I'm using histology to see if, we're creating any sort of damage to tissue or causing any, inflammation through sort of physical damage. We could also sort of look at, microbes infecting, some of the host cells in that way. We've been looking at C-reactive protein in the blood to make sure we haven't elevated systemic levels of inflammation. And last but not least, we're also sort of looking at the gut microbiome. Now, if we shifted the gut microbiome, that might be a really positive thing, but we need to make sure that we're doing this in a positive way or to see, maybe there isn't any impact, and that might be great as well. But we need to certainly assess that. Preliminary evidence suggests that it is safe. We haven't seen any evidence in our data so far that there's signs of inflammation, so that's really promising. There's also been two other OMT studies that have been completed. One was in dogs with some periodontitis in dogs, and their setup was very, very different. And there's been no sort of in vitro growth and testing and selection for donors and things like that. But in both of those publications, they were able to show that it was safe. So in both cases, the dogs didn't get secondary infections, or in the other study where they were looking at mice, there was no signs of inflammation and no signs of secondary infection. So just as we see with fecal microbiome transplantation or FMT, that's done with the gut microbiome, we think that it can be safe, but obviously there's going to be risks involved. So in the fecal microbiome world, some of the early studies, they were giving a fecal microbiome transplant to cure one thing, and then they sort of had knock-on diseases that happened later. And the classic one was one that was done between siblings, and it actually a transplantation happened from an obese sibling to a lean sibling, and it caused that lean sibling to become obese. It also treated the other disease that they were trying to treat, but we also sort of had that knock-on effect of obesity. So there's certainly these sort of secondary effects, which we are highly aware of, but honestly will take further investigation. And it's hard to predict what those might be. But I think that, it's worth it to sort of push forward and see, but absolutely something we're going to have to look at. I think the best way we're going to try to tackle it is by looking in rat and mouse models of systemic disease and seeing whether or not OMT makes those diseases worse or better and trying to really sort of get at it using these mouse models so that we're not harming humans.
Mhm. I'm glad you brought up the fecal transplants because that's a really good parallel because you have this precedent of safety and some side effects here or there that you can work through, with whether that be improved screening of the donor microbiomes or whatever that might be. But I think most of the side effects now are just what bloating and constipation and things like that for fecal. So hopefully for the OMT world, the side effects will be very, very minor, maybe slight stippling of the gingiva and you just have to I don't know what that could look like, but hopefully that's a similar model there as well.
Yeah, we really think so. And that's why we're really making sure we're selecting from ultra healthy donors, right? We want to take these microorganisms for people who don't have obesity or don't suffer from colon cancer, right? And so we're really trying to again, look at systemic disease in our ultra healthy donors. It's not just that they're orally healthy, they're also systemically healthy, when we're
Right.
selecting them. So that's really important.
Awesome. Well, I guess now we can talk about how our audience can get involved in this exciting project. And so if you all who are listening and or watching want to be part of this revolutionary research, and provide your own feedback, we would love for you to participate. And that's whether you're a student, a patient, a dentist, a specialist, a faculty member, basically any of you. You can participate in a quick 5 to 10 minute interview and let your voice be heard and share your thoughts about this project with Laura, Kevin, and the rest of the team. And so the interviews are open from February 12th through March 22nd of this year. And you can sign up a few different ways. You can send an email to oralmicrobiometransplant@gmail.com. I'll leave that email address in the description of this video. Or you can click on a link that I will also include in the description that'll bring you to a calendar that you can directly sign up there. And one more way, we'll put a QR code on the screen right now, which you can scan with your camera app on your phone, and that will also bring you to the calendar where you can sign up to do an interview. And then one more thing, next month in February, on Thursday the 15th at 7 p.m. Eastern Time, we will be returning. This time we will be live. So we'll do another live event, which we've done on the channel before. And so if you have questions that you want answered, Laura, Kevin, and I will be here and you can ask away.
Awesome. Yeah, Kevin and I would love to speak with you about our project and how you think we should develop in the future or commercialize this technology. And thanks so much for having us on your podcast. We're so grateful to be here. Thanks so much.
You're welcome. Thank you. It was a lot of fun, and we'll talk again soon.
Thanks, Ryan. Cheers.